All right, everybody. Now we are going to be modeling cellular respiration in a plant cell. So I'll go ahead and start with the directions that it lists. Step number 1 is color all the mitochondria in pink. Remember, our mitochondria typically are drawn with this kind of little squiggly line here. So I'll go ahead and draw those, color those in pink. Step number 2 says color all the cytoplasm in light blue. This is a little bit tough, so bear with me here. We've got our cytoplasm kind of headed around all of these objects. It's that space that fills up everything in between all of our organelles here. So we color all of that in blue as neatly and quickly as we can. Going kind of around some organelles here that we aren't super familiar with. We've got endoplasmic reticulum, we've got vacuoles, we've got nuclei, well, one nucleus, some things like that. Excuse my messy coloring here, please. Okay, so there's our cytoplasm colored in. Next, it says that we have to draw an arrow showing glucose entering the cell. I am going to go ahead and grab my pink marker here, right like that. So I'm going to go ahead and I'm going to draw glucose. I'm going to go right over here. So entering the cell, we have glucose, and it also asks us to state the type of transport and where that glucose is coming from. So in this case, we know that glucose is always too big to enter the cell on its own, so it has to use facilitated diffusion. Facilitated diffusion. And it tells us that this glucose is coming from the stem. Let's change that. We'll say from stem. So I go ahead and add that myself. Okay. Once glucose is in the cell, now it can break apart into smaller molecules so that it can be used inside specific organelles in our cell. So I will go ahead and pick my glucose color. Remember, this should look very similar to what it did in our last image. 2, 3, 4, 5, 6 is glucose. We take that and we break that into 2 pyruvates, which look like this: 1, 2, 3, and 1, 2, 3. And so that is glucose into pyruvate. So I'll put glucose, let's make it a bit smaller. Glucose into 2 pyruvates. Okay. Now we have to draw an arrow showing pyruvate entering the mitochondria. I'm going to just circle it in gray so I could visually see my circle here. My pyruvates are going to enter the mitochondria here. We also have some other things that are entering and leaving the cell at this point. So we have, it mentions that we have to draw an arrow showing oxygen entering the cell and the mitochondria. So I'm going to draw from the outside environment into my cell. This is my oxygen, and it would like us to state the transport movement and that it's coming from the outside environment. So my transport movement for oxygen is always going to be diffusion. It's small enough to make it through that phospholipid bilayer, and oxygen for a plant is always coming from the outside environment. It's not an M. Here we go. So from the outside environment. Okay, so we've got our inputs here. Step number 6 says that we need to draw an arrow showing water exiting the mitochondria and the cell. So I'm going to draw, let's see, I'm going to kind of go this way. Notice that we're not actually going through the vacuole. I'm just drawing over the vacuole. Our water here is exiting the mitochondria. And actually, you know what I can do here? So this isn't confusing. I'm going to draw my arrow headed out this way. My water, and The water in this case has a transport movement. Remember, water is like diffusion, but we call it osmosis because it's in particular for water. Okay, so our water exits here. And last but not least, we need to use an arrow to draw carbon dioxide exiting the mitochondria and the cell. So again, I'm going to go around that vacuole straight out of the cell. We have carbon dioxide. Carbon dioxide is one of those molecules that is small enough to pass through the membrane. So this is going to be diffusion for the transport. And it says that this is going to the outside environment. So I write that as well. Okay. Lastly, we have to go ahead and make our ATP. That's a pretty important part of this process. It's actually the whole point of this process. So I'm going to go ahead and out of my mitochondria, I'm going to draw 36 ATP that are produced. And again, I'm going to kind of outline that because I know that my 36 ATP is the whole point of this process. It's the whole point of doing cell respiration. We need that energy. Another reminder here, we are able to do this because we have oxygen present over here on the right-hand side. If oxygen were not present, that glucose here would not enter the mitochondria. It would stay in the cytoplasm and it would do a type of fermentation that is specifically for plant cells. We call that fermentation here. Okay. And that is all we've got.